Hello everyone, I hope you are all good. In last class we have discussed about Hershey and Chase experiment from which it is unequivocally proven that DNA is the genetic material in organisms. Now let us discuss the properties of a genetic material. Here we will be focusing on DNA versus RNA. In some virus, RNA act as genetic material. For example, tobacco mosaic virus, bacteriophage, retrovirus, which include HIV and even common cold virus, that is rhinovirus. But DNA is predominantly a genetic material in majority of the living organisms, whereas RNA will be in some virus. Now you can ask one question, whether the organisms which are having DNA as their genetic material, whether they have RNA or not. Of course, the answer will be yes. RNA is the RNA will be performing other dynamic functions such as it act as messenger and it act as adapter during the process of protein synthesis. Even it act as a catalyst for various types of activities in a cell. Now, DNA and RNA. Among these, DNA is predominantly a genetic material, and our question will be why? Why not RNA will be the genetic material in majority of living organisms? Why DNA is predominantly a genetic material? To understand this, first we should know the two important chemical differences between DNA and RNA. In case of DNA, there is presence of deoxyribose sugar. The sugar, that is pentose sugar, deoxyribose sugar, will lack one oxygen at second carbon, and the nitrogenous bases present in DNA are adenine, guanine, thymine and cytosine. Whereas in RNA, the ribose sugar is present, that is the pentose sugar present is ribose, and that have the presence of OH group at second carbon. And very importantly, the nitrogenous bases found are adenine, guanine, uracil and cytosine. Here you have to make out the difference: thymine and uracil, OH group and H at second carbon. Yes, these two are the two important chemical differences between DNA and RNA. This you should remember. This is very, very important to understand the further topic. Now, let us come to our actual topic. To call any molecule, that could be protein, DNA or RNA, to act as a genetic material, it should fulfill four important criteria. And those four important criteria, let us discuss one by one. The first criteria is it should be able to generate its replica. That is, it should able to undergo the process called replication. That is, it should produce multiple copies of its. Now, among protein, DNA and RNA, in case of DNA and RNA, there is a rule of complementarity. That is, purines will base pairs with pyrimidines, adenine with thymine, guanine with cytosine. Now, due to this complementarity, DNA and RNA are able to produce their replica. That is, both DNA and RNA will fulfill the first criterion, whereas protein is a polymer of amino acids and there is no rule of complementarity and hence there is no replication. Now, from our list, protein is deleted. Now, let us go for the second criteria. The molecule should be stable chemically and structurally. That is, it should not undergo any changes with stages of life cycle. Secondly, with the age and the physiological functions of an organism. Within an organism, any of these should not alter the structural stability and chemical stability of a molecule that is acting as a genetic material. Now, in our list, there are only two, DNA and RNA. From Griffith experiment, we have studied that after heat killing of the S strain, something is remained. We called it as a transforming principle. Now we know that that is DNA. That is, it need not undergo the process of destruction. So that is heat killing. That is, heat will also not destroy DNA, means it is stable. Then you can ask question, nothing happened to DNA? Of course, upon heating, the double-stranded DNA as converted into single strand due to the breakage of hydrogen bonds. Now, again, due to complementarity, the two strands will again form a double strand under the suitable conditions. So this is a very fascinating process which happen in double-stranded DNA. For this, there are reasons. The first reason is the presence of adenine and thymine, guanine and cytosine have presence of hydrogen bond, that is complementarity. Secondly, the stability is due to the deoxyribose sugar, and deoxyribose sugar lack OH group at second carbon atom, and presence of thymine in place of uracil will also give additional stability to DNA. Whereas in case of RNA, at second carbon there is presence of OH group, and this OH group is highly reactive, that is replaceable. Any molecule can replace OH group easily, not H. So the RNA have the presence of OH group at every nucleotide, hence it is reactive, and hence it is liable. Liable means easily undergo any changes, that is called easily undergo alteration, and hence it can undergo degradation. But that is DNA cannot undergo degradation easily because of the reasons we discussed. Also, RNA, being it is catalytic, it is highly reactive. This is important, very important point you should remember. Now, from this point, that is second criteria is fulfilled completely by DNA. Hence, DNA is a better genetic material as compared to RNA. So that is, in some virus, RNA will be the genetic material, but better genetic material is DNA. The third criteria is the molecule should provide scope for slow changes, and those slow changes are called mutations, and the mutations are required for evolution. Now, again, in our list there are two, DNA and RNA. DNA is able to mutate at a slower rate. Being it is stable, it will mutate at the slower rate. Whereas RNA, being it is unstable. Unstable means it is highly reactive. Highly reactive means it will be unstable, mutate at a faster rate. Consequently, those organisms which are having RNA as their genetic material, for example, virus, will have a shorter lifespan and mutate and evolve at a faster rate. So this is very important point. Those having RNA as a genetic material, they have shorter lifespan. So now you can connect the thing why majority of living organisms have DNA as their genetic material, because they want to live more. Now, since they mutate at a faster rate, they produces more number of their strains. For example, the fresh example is COVID virus, which has produced many variants in a short lifespan. Now, the fourth and last criteria, it should be able to express itself in the form of Mendelian characters. So, expression in the form of Mendelian character is nothing but protein synthesis. So, any organism, character expression is nothing but protein synthesized. If whatever the protein synthesized, based upon that, character is decided. Now, RNA can directly code for the protein. That means what? It can express easily. Easily it can express, whereas DNA will produces mRNA, that is intermediate. That means it choose RNA intermediate for expression of the character. From that mRNA there is production of protein. That is, it is one point is clear, the protein synthesizing machinery in case of organisms having DNA as their genetic material has evolved around RNA. So very first I told, those organisms which are having DNA as their genetic material will also have RNA, which are having other functions. So now DNA and RNA, both of them can function as a genetic material. We have studied they both can replicate, both can express in the form of Mendelian characters. So both of them can act as a genetic material. But being DNA is stable, it is a best candidate for storage of genetic information. Whereas RNA is a better candidate for the transmission of genetic information. That is, transmission means from DNA, the information is transmitted to mRNA. From mRNA, into protein. So by that way the expression of character will takes place. Now from this ongoing discussion there are two questions answered: why DNA is a predominant genetic material, and second question, criteria for a molecule to call as a genetic material. So this topic is very very important. In next class I'll come up with a very interesting video, so stay tuned and don't forget to subscribe our channel. Thank you.